Today we have the defense of the candidate, Mr. Sohel, and it's the second step in the examination of the PhD progress. Today we have the oral presentation, and the first step was the written examination of the PhD thesis. So it was today, I think it was a positive situation that we can continue the examination today, and I have to give some information to you about the following, let's say, 1.5 hours. And after that, I will introduce the candidate, the person who is today at the examination. The regulations are written down from the Dean Office, and it's so that in the first 30 minutes we have the oral presentation of the candidate. After that, 15 minutes you all have the possibility to ask questions, and then we make the following, let's say, examination part non-public, so only the reviewers, Professor Schellander and Professor Sülekum, with me and the candidate, we have the possibility to ask questions without the public possibilities. So after an additional 30 minutes or until 1 hour, the examination is finished, and then we can give all to you the result of the total examination. This is, let's say, the regulation part, and I hope everything will done successfully today. Now it's up to me to introduce the candidate. Mr. Sohel made his bachelor work in Bangladesh at the Agricultural University from the year 2001 to 2006. After that, he continued his academic work also in the same university with the Master in Animal Breeding and Genetics. And since 2010, he's working here at the Institute of Animal Science, Bonn University, and works about, I think, this topic of the presentation today. And now it's up to you to start your presentation. You try to keep 30 minutes. I think you will do that. Thank you.
Thank you, Honorable Chairperson. Ladies and gentlemen, good afternoon. I would like to welcome you all in my PhD defense presentation titled Exosomal and Non-Exosomal Circulatory MicroRNAs in Bovine Follicular Fluid. Potential role of exosomal microRNAs in oocyte development. The primary goal of livestock rearing is to get the consumable products from the animal, for example, meat, milk, etc. The productivity of animal can be enhanced through better reproduction and breeding management. Unfortunately, there is a negative correlation between productivity and fertility. For example, increase of each 100 kg of milk production, there is a 6% loss of pregnancy and 8% loss of ovarian activities. The loss of fertility in high-yielding dairy cows may be due to negative energy balance during the last phase of pregnancy and the early phase of lactation, which is characterized by disturbed folliculogenesis, early embryonic loss, abortion in late pregnancy. Among those, disturbed folliculogenesis and early embryonic loss comprises 70 to 80% loss, indicating that still we have huge opportunity to increase the fertility, focusing on this area. In order to increase fertility, thorough investigation in molecular level is essential during the process of folliculogenesis or follicular development. So, follicular development is a complex coordinated process which involves morphological changes in follicular cells from flattened unilaminar cells to cuboidal unilaminar cells. These cuboidal unilaminar cells differentiate into thecal cell and multilaminar granular cells, These granulose cells undergo quick proliferation and apoptotic process to facilitate antrum formation and as well as follicular fluid formation. Extensive cell-to-cell communication within somatic cells and somatic gonadal cell is essential to have a successful follicular development. Different molecular pathways, like including pro-apoptotic pathways and anti-apoptotic pathways, are involved in this process. At the end of this folliculogenesis, a developmentally competent oocyte is released for fertilization. Oocyte developmental competence can be defined as the ability of an oocyte to resume meiosis, to give follicle fertilization, to develop to the blastocyst stage, to induce a pregnancy and bearing offspring, to term with a good health. So far, scientists have developed several methods to assess the quality of an oocyte, including non-invasive like morphological assessment or the use of brilliant result blue stain, or invasive method which is a molecular level, in particular transcripts in tubular cells, granulose cells and oocyte. Studies have shown that a fully grown oocyte selected by using a brilliant crystal blue shows better performance during in vitro fertilization compared to a growing oocyte. It has been shown that several genes which are upregulated in several stages of follicular development. It means that there is a switch on, switch off mechanism of those genes. Which molecule involved to make them switch on or switch off? So it is well recognized that microRNAs are the major regulator of gene expression. So several studies also showed that some microRNAs are overexpressed in different stages of follicular development. It clearly indicates that maybe these microRNAs are involved in this developmental process. So far, in our group also find a different set of differentially regulated microRNAs in human cell by comparing a mature oocyte and an immature oocyte. While other studies also find some differentially regulated microRNAs in granulose cells as well. However, it is completely unknown either microRNAs are present in follicular fluid or do they perform any function. While majority of microRNAs are found intracellularly, a handful number of microRNAs are also detected in outside the cells, which is commonly termed as extracellular microRNAs or circulating microRNAs. Like cellular microRNAs, the circulating microRNAs are 18 to 22 nucleotide long, but detected in different biofluids. Unlike cellular microRNA, circulating microRNAs are remarkably stable in outside the cellular environment, where the presence of high RNA activity. They are resistant to harsh conditions like boiling, low or high pH. They can be stored for a long time, and they are rarely detected after multiple freezing or thaw cycle. As because they can satisfy several aspects of a successful biomarker, they are frequently used as a biomarker for different altered physiological and pathological conditions. The mechanism underlying their remarkable stability in extracellular environment suggests they might wrap with some protective mechanism, and this wrapping hypothesis also offered us some release mechanism of these microRNAs. So far, studies in different biofluids shows that microRNAs can be released in the extracellular environment via shedding vessels like exosomes, microparticles, or apoptotic bodies. Or they can be first coupled with protein complex or high density lipoprotein and then released to the extracellular environment. Among those pathways, exosome-mediated release of extracellular microRNAs got the research priority because of their viability. Exosomes can be released by a viable cell and uptaken as well. However, the biological viability of other mechanism is still unknown. So here is a hypothetical presentation how the exosome, one cell, can communicate with other cell by exosomes. First, microRNA can be selectively incorporated with the intraluminal vesicle of a multivascular bodies, and then fuse with this multivascular bodies fuse with the cell membrane and release the intraluminal vesicles as exosome. These exosomes can travel a long distance and bind with the surface of a recipient cells by receptor-ligand docking system. And then they can directly fuse with the plasma membrane, or they first be endocytosed and then fuse with the endocytic membrane and finally release the microRNAs into the cytoplasm of the recipient cell. In both cases, this microRNA may be associated with the silencing of corresponding messenger RNA. So the research gap is presence of extracellular microRNAs in bovine follicular fluid, their potential association with oocyte developmental competence, and their role is currently unknown. Accordingly, we hypothesize that first, extracellular microRNAs are present in follicular fluid and their abundance can vary depending on the growth status of the oocyte. Secondly, extracellular microRNAs can be taken up by the surrounding granulosa cells and play a role in cell-to-cell communication during follicular development. Our objectives are to identify and characterize the mode of circulation of extracellular microRNAs in follicular fluid, to investigate the extracellular microRNAs signatures in follicular fluid derived from follicles containing a growing oocyte or a fully grown oocyte, to demonstrate exosome-mediated transfer of microRNAs in follicular microenvironment. To achieve those objectives, we divide our whole study into two experiments, where the first experiment focused on the identification and characterization of extracellular microRNAs in follicular fluid, and the second experiment dealing with the functional delivery of microRNAs via exosome in follicular microenvironment. Identification of extracellular microRNAs in bovine follicular fluid. Ovaries were collected from the local swarm house irrespective of estrous cycle or presence or absence of corpus luteum. On an average, 4 to 8 millimeter follicles were bland dissected and the oocytes were retried. The oocytes are subjected to brilliant crystal blue staining to differentiate or to identify, categorize them as a growing oocyte or a fully grown oocyte. According to the BCD staining of the oocyte, the follicular fluid and the surrounding cells were collected. On an average, a total of 60 follicles were used in each group, where 20 follicles were used in each biological replication. Follicles from both groups were purified using centrifugation and filtration. Exosomes were isolated from purified follicular fluid using exotic precipitation or autocentrifugation. And then exosomes, the efficiency of exosomes isolation was validated using Western blot and electron microscopy. Following total RNA isolation, CDN was synthesized and microRNA UP cell array was used to identify those microRNAs and find out the differential regulation of the microRNAs. Data was analyzed using available web-based analytical softwares. First, we characterize the efficiency of exosome separation in protein level, where 1 and 2 represent exosomes from exotic precipitation or autocentrifugation.